These are the Unit 7 notes, Cell Cycle Part 3. In this particular video, we're going to be reviewing the cell cycle and going through the different series of steps that a cell would go through in order to complete the cell cycle. So let's start off at the top where it says cell cycle. We're going to write each cell goes through a series of steps to grow, develop, and divide. That means we're going to be making a new cell. In other words, we're going to be going from one cell and making two new cells. Where it says the two main stages of the cell cycle, the cell is going to be going through the two main phases called interphase and M phase, or the mitotic phase or mitosis phase. Interphase is going to last the longest, and interphase is going to prep the cell for M phase. And we will discuss those steps down below. The next stage is called the mitosis or mitotic phase. This is when the cell actually divides. So the cell divides into two new cells. So this is where we actually see the cell dividing into those two distinct cells. Now let's go down below where we see this circle. So if I were you, I would pause the video right now and I would draw this particular picture. So I would make a large circle, right? Go all the way around and then split it up and do the picture as best you can. Now, once you pause the video and you make this diagram, I'd make it pretty big. Okay, this is probably one of the main things here on this paper. And then you're going to record the information inside of this specific circle. All right, so I'm going to start off by labeling this outside part right here. This is called interphase, which is what I mentioned above. So interphase is going to be the longest part of the cell cycle. And interphase is going to be divided up into three phases. We have what's called the G1 phase, the S phase, and the G2 phase. Let's talk about the G1 phase first. During this particular phase, the cell is going to grow larger, or it's going to get bigger. It is also going to copy organelles or make more organelles to prepare itself for cell division, which is the mitosis stage. After it passes the G1 phase, it's going to enter the S phase. During the S phase, the cell synthesizes, that's what the S stands for, or makes a copy of its DNA. And the reason it does this is because we have to make sure that each new cell is going to get the correct amount of DNA when that cell splits into two cells. So if we start with a cell of 46 chromosomes, we need to end with two identical cells that also have 46 chromosomes by the very end. After it passes through the S phase, it's going to go on to the G2 phase. This is where the cell grows more. So I like to think of the G as growth. It also prepares itself for mitosis, which is cell division. So again, we have interphase, which consists of G1, S, and G2. Now that we've talked about interphase, the cell will then enter what's called the M phase or the mitosis phase. So now let's zoom in over here. We're going to make a bracket around this area. This is M phase. Now, specifically, these 4 steps are going to be called mitosis. Now, mitosis, we'll get into more either later on this week, probably into next week. We're going to talk about these specific steps in more detail. But the 4 stages are called prophase. So P for prophase, M for metaphase, A for anaphase, and T for telophase. So I like to think of these as PMAT. So PMAT always helps me remember the order of the steps. So I'm highlighting P, M, A, T. And like I said, we will discuss those more in detail, but that is what's going to get the cell from 1 cell to 2 cells. So this is the actual cell division part. And then the last step of in phase, this is going to be called cytokinesis. So cytokinesis, you can see, is not actually a part of mitosis, but that is going to be when the cell actually fully splits into 2 cells. It's just that kind of final step of the in phase. As you can see, interphase is going to take up a majority of that cell's life cycle. So you can see it goes around about 3/4 of the way. When we take a look at M phase is this smaller chunk right here. So a majority of time will be spent in interphase, and then the cell will go through M phase pretty quick. Now, a typical human cell, this process takes about 24 hours to divide. In smaller things like yeast cells, this might take 90 minutes to fully get through, or in bacterial cells, it might only take 20 minutes to get through. So it really just depends on the living thing and what type of cell it is. Now let's go on to the last thing for notes. Down at the bottom where it says apoptosis. Apoptosis is programmed cell death. During apoptosis, cells are going to get a signal to shut itself off. In other words, the cell is going to die. It's no longer going to function. When apoptosis happens, it cannot come back. Essentially, apoptosis is just a general way or a convenient way for the body to remove cells that should no longer be a part of the organism. Now, you have cells that are called cancer cells. The special thing about cancer cells is that they do not go through apoptosis. In other words, they keep dividing over and over again. In other words, because they don't go through apoptosis, the cell is going to not shut itself off. In other words, it's going to stay on, and it's going to keep dividing over and over again and constantly going through those different stages of the cell cycle to make more of itself.